Good afternoon. Welcome to the NCOH monthly webinar. My name is Martin Hoek. I'm the communication manager for NCOH, and I will be your host today. This meeting will be recorded and made available through the NCOH website. You can also view our previous webinars through that link. This webinar will be, we have a special guest called Rebecca Osborne, and she will make a graphic report of this webinar. That will also be available. This month, we will have presentations on antimicrobial resistance, since this is the week of the World Antimicrobial Awareness Week. The purpose of this week is to increase awareness of global antimicrobial resistance and to encourage best practices among the general public, health workers and policymakers to avoid further emergence and spread of drug-resistant infections. Antimicrobial resistance, or AMR, occurs when bacteria, viruses, fungi, and parasites resist the effects of medications, making common infections harder to treat and increasing the risk of disease spread, severe illness, and death. Antimicrobials are agents that are critical tools for fighting disease in humans, animals, and plants, and include antibiotic, antiviral, antifungal, and antiprotozoal medicines. Multiple factors, including overuse of medicines in humans, livestock, and agriculture, as well as poor access to clean water, sanitation, and hygiene, have accelerated the threat of antimicrobial resistance worldwide. Now, before I give the word to our guest today, just a reminder that you can ask questions using the chat function. We will use those questions later in the discussions with the presenters. So let's start with the presentations. Our first speaker of today is Professor Nathaniel Martin. He is a full professor of biology chemistry at Leiden University. He obtained his PhD at the University of Alberta and his postdoc at UC University College of Berkeley. He also managed to learn Dutch to level B2. I think that's better than mine, actually. Today, he will tell us more on the novel glycopeptide antibiotics with best-in-class potential. Professor Nathaniel, the floor is all yours.
Thank you very much, Martin. And I will begin by saying I'm much more comfortable doing this in English, so it's still going to be a while before I have overcome confidence issues related to presenting in Dutch. It may will happen one day, but today's short lecture will indeed be in English and will specifically touch on a new class of semi-synthetic antibiotics that we've developed in the last couple of years in my lab, known as the guanidino lipoglycopeptides. And we think that these compounds may in fact have best-in-class potential based on some of the results I'll show you today. Now, you'll hear a lot of people talk about how nowadays the big needs in anti-infectives are, of course, for gram-negative pathogens, and that's true. We have some very, very difficult, high-priority gram-negative pathogens that we don't have a lot of good treatments for due to drug resistance. However, the pathogens you see here, MRSA and VRE, do currently account for about half of all AMR-associated deaths in the United States and Europe. And so one can't underestimate the importance of having effective therapies to treat these types of gram-positive pathogens as well. And that's the type of compound class that I'll present today. We are also in my lab, of course, working with anti-gram-negative agents, but today I'll specifically talk about a class of compounds that has very potent activity against gram-positive pathogens. And the story starts with a bit of background, namely into the glycopeptide class of antibiotics, typified by the molecule you see here, vancomycin. Vancomycin was introduced into the clinic in the 70s, and since that time has been used frequently for the treatment of infections due to Staphylococci and Enterococci. And it's been used very successfully for many years. However, resistance is certainly widespread nowadays to vancomycin. Certainly, if one thinks about the rates of VRE, in some cases, they're actually the dominant enterococcal infections encountered in hospital settings. And there's actually not that much of a discussion, but it's certainly known if you talk to clinicians about the toxicity concerns surrounding the use of vancomycin. And so I'll touch on that a little bit today too. But first of all, as I said, resistance to vancomycin is quite widespread, and this was already the case as early as 20 years ago. And that led to the generation of semi-synthetic analogs known as the lipoglycopeptide antibiotics. And you see an example of that molecule, or of that class here, the molecule telavancin. Telavancin is in fact generated starting from vancomycin by organic chemistry. You modify it to include a tail, a lipid tail here, and another modification near the C-terminus. And telavancin does have enhanced activity relative to vancomycin. It works relatively well against some VRE strains, but it also has increased renal toxicity and very unusual pharmacokinetic properties. In fact, the half-life of these types of lipoglycopeptides can be as high as 300 hours. So that's, depending on how you want to frame it, either an advantage or a disadvantage. The advantage may well be that a patient can be dosed and then sent home where they've got the drug circulating in their body for a week without the need to re-administer. However, if you need to stop giving this drug to a patient, if there's a poor response, it's very difficult then to do anything about it, given those PK properties. So this combined, as I say, with the increased kidney toxicity of telavancin actually has led to it being slapped with a black box warning from the FDA. So with this background, we decided to see if we could do something to try and find the sweet spot, say between enhanced activity but maybe reduced toxicity with a new class of semi-synthetic glycopeptides, and those are the guanidino lipoglycopeptides, as we're calling them. And they're called guanidino because they contain a guanidine group, which I'll show you in a second. But this is something that our lab is has quite a lot of expertise in incorporating guanidine groups into biologically active molecules, peptides, small molecules. And why is the guanidine group interesting? Well, it contains a very strong positive charge. In fact, amongst all the amino acids, it's the amino acid arginine that contains a guanidine group. This is almost entirely charged at physiological pH. And so we had the idea, using some chemistry that we developed over the years, to try and introduce a guanidine group into a lipoglycopeptide. And that led us to the class of compounds you see here. So again, the vancomycin core, and now we've modified the vancomycin to contain both a guanidine group, and then linked to the guanidine group are lipids. And we can vary the nature of these lipids. We've made dozens of compounds with this general structure here. And they're very straightforward to prepare. The yields are very good. It's scalable and cost effective, given that you're starting with vancomycin, a natural product, as the starting material. And what I'll show you on the next slide, and actually for the rest of the talk, is data from one of our most promising analogs from this class of compounds. And here you see, first of all, some in vitro activity associated with these compounds. And here I'm comparing on the left vancomycin with one of our guanidino lipoglycopeptides on the right. And below you see the in vitro MIC values. So these are the minimum inhibitory concentrations required to kill the strains indicated. And so you can see for things like methicillin susceptible or methicillin resistant Staph aureus, vancomycin has a reasonable MIC of 1 microgram per mil. This is certainly clinically useful. However, look at the compounds that we're working with. We see, like in some cases, almost thousands of fold increase in activity. And it gets really interesting when you start to look at the vancomycin intermediate and resistant organisms. So vancomycin resistant Staph aureus, VREs, you can see generally quite resistant organisms. And then again, if you look over the activity of these next generation guanidino lipoglycopeptides, you see they maintain their potent activity across the board. So very interesting antibacterial effects. But of course, before you get too excited with these types of results, you need to also confirm that this is bacterial specific, that these compounds are not just serving as membrane disruptors, that introducing this group hasn't just made the compounds a more aspecific cell membrane disruptor. And so to do that, you can use some cell-based assays. Most notably, the first thing you do is check to see what the effect of these compounds is on red blood cells. And we are happy to see that the guanidino lipoglycopeptides are very non-hemolytic, up to 1,000-fold the killing concentration. And then also we've done some preliminary studies with kidney cells and shown that the compound indicated here, our guanidino lipoglycopeptide, shows no appreciable toxicity up to 100 micromolar concentration. Neither does vancomycin, notably. But also of interest, we included some clinically used glycopeptides, telavancin and oritavancin. And in fact, they are quite toxic to kidney cells. And I should say, both of these are next generation glycopeptides that are used in hospitals today. So interesting to see that there's a certain level of toxicity already built into some of these clinically used drugs that we seem to be able to circumvent. So that's promising and suggests, in fact, that this potent activity we're seeing is specific to the bacterial cell and is not a general membrane disruption activity. The next set of assays we did was aimed at assessing whether or not these new compounds were more or less likely to induce resistance. And so an assay was done called a serial passage assay. And here we started with a sensitive strain of MRSA that we could kill with a relatively low concentration of our compound. We took daptomycin along as a reference antibiotic. And this assay basically involves a sequential passaging of the organism in the presence of a slightly less than lethal concentration of antibiotic. And when you do this, in many cases, bugs will adapt and eventually require higher concentrations of the antibiotic to be killed. And you certainly see that's the case with daptomycin. Over the 30-day course of this experiment, the MIC for daptomycin rises from 1 up to 16. And so this is no longer sensitive, obviously. Whereas in green, you see the sort of trend for our compound, very little, if any, real change in sensitivity over the 30-day course of the experiment. So, and also big props to the PhD student, Emma, who did this experiment. This involves exactly what you might assume, and that is the PhD student comes in every day for 30 days, weekends as well. So it's a real dedication to getting this kind of result. So kudos to Emma. So with these promising in vitro data and resistance studies, we then moved on to actually evaluate these compounds in vivo in an established mouse model. The first thing we did was just to see how well the compounds are tolerated. And the compound that we took forward into mice was very well tolerated up at the highest dose tested of 100 mg per kg subcutaneous. And also of note were the favorable pharmacokinetics that we saw for this compound. So a clearance time, half-life of about 1.2, 1.23 hours. And remember, this is maybe of relevance considering that some of these other clinically used lipoglycopeptides have these ridiculous 200 and 300 hour half-lives. So that seems to be favorable. And when we dosed this compound at 3 mg per kg, we were able to ascertain that we actually maintained concentration in the bloodstream above MIC for more than 8 hours. So also looks to have good properties that way. So we proceeded then to an efficacy study, sort of the industry standard, gold standard in this case is a neutropenic murine thigh infection model. So in this case, mice with a sort of suppressed immune system are infected in the thigh muscle using MRSA bacteria. And here you see in the group of mice that were not treated with antibiotic, over the 24-hour course of the experiment, the number of bacterial colonies counted in the thigh muscle over that 24-hour period dramatically increases. In orange is the group that was treated with vancomycin at 25 mg per kg every 12 hours. And then in light and dark green are the groups of mice that received our compound. And you can see with as little as 3 mg per kg dosing every 6 hours, so for a cumulative dose of 12 mg per kg versus 50, you already see a significant decrease relative to vancomycin. And then when you go up to a 10 mg per kg dosing of our compound every 6 hours, you see essentially clearing of the infection just near the limit of detection in the assay. So this is quite compelling, I would say, demonstrating that these compounds are significantly more effective than vancomycin in vivo, and nicely mirroring our in vitro data. So the next steps, well, we've demonstrated, at least in some preliminary assays, that the guanidino lipoglycopeptides do outperform vancomycin and some other clinically used glycopeptides. And for that reason, we were wondering, maybe have we got something that has a best-in-class potential? And that, of course, relates to not only their enhanced activity, but if the lower toxicity is indeed a true effect that we can see in further advanced assays, we could be looking at a class of compounds that has a greater therapeutic window, which is, of course, very important. To further establish this, we were recently awarded a grant from the Dutch Science Foundation under the NACTAR fund. And this is funds specifically aimed at actually supporting preclinical drug development efforts. So here you see on the left side a number of assays that we'll be doing in the next 2 years to really see how safe and efficient or effective are these new compounds. So that's exciting stuff. We just heard that we got this grant a couple of weeks ago, so we're really excited to move forward with these compounds. With respect to the IP landscape, well, there's a lot of work that has been done, of course, in the glycopeptide field. We filed a patent covering our guanidino lipoglycopeptides about a year ago, and about 2 months ago got the examiner's report, and we were very pleased to see that the examiner had assessed all of our claims as both novel and inventive. So that's very important. And the PCT filing is now underway. We're looking at taking these compounds forward with an experienced team, ourselves, of course, doing the chemistry and biochemistry. Evotech is a CRO that we work with that they're experts in the infection models, the in vivo work. And then Of course, on the business development side, we've got the tech transfer office in Leiden, as well as the patent attorneys that are supporting the effort. I think just to close, it's important to, again, think broadly and look forward for where these compounds might fit into the clinic. I would say there is certainly a need and a market for a safer and more effective glycopeptide antibiotic. In talking with Mark Bonten, who many of you will know, he underscores the importance of developing new compounds to address VRE, certainly in hospital settings. And then another colleague of mine at the Erasmus MC, he's a pediatric nephrologist. Actually, in talking to him, I was surprised to find out that often when they see kids with kidney injury, it's linked to a suspected vancomycin treatment. So again, this gets to this point that I raised earlier, that the toxicity of vancomycin isn't probably fully appreciated, but it's something that people in the clinic do encounter. And so if we can develop a glycopeptide antibiotic with lower kidney toxicity, that would obviously be very, very favorable. And so to that end, we, as I say, are excited to start these more advanced preclinical studies to establish just how good these compounds are and whether or not they've got clinical potential. And so with that, I'll wrap up with the acknowledgement slide. Here you see the people in our group in Leiden, as well as some key collaborators that contributed, the funders. And then I want to raise special attention to the three folks here with the stars beside their name, Emma, Nicola, and Kamal. The three of them were very instrumental in getting this project off the ground and essentially did all of the work in terms of synthesizing and doing the in vitro testing. So big props to them. And I'd like to thank you for your attention. I'm happy to take questions.
Thank you, Professor Martin, for this informative talk. We have a few questions in the chat function, but before we go there, I have a question to you regarding slide 8 in resistance studies, where you compare Quantino, and how quickly the bacteria build up a form of resistance. And it seems that when the new drug that you developed, it's much slower. And I was wondering if you could tell us more about the mechanisms behind it, and does this mean that there will never be any resistance for this drug, or how does that work?
Yeah, I mean, that's a good question. You indeed want to understand the mechanism of something if it's doing something that appears to be new or better. Yeah. And we think that introducing the guanidino lipid function into the antibiotic is leading to an enhanced binding to the bacterial cell surface. The strong positive charge of the guanidine group very likely drives electrostatic interactions with the negatively charged cell surface. And you say something very perceptive, and that is, why is it that the bacteria can't apparently become resistant? Well, I think the bacterial cell, in trying to adapt, would need to severely reduce its overall negative charge. And that's something that, well, it can be done, is not something that happens very quickly or in a very stable manner. And so I think that, yeah, you hesitate to ever talk about something being resistance proof, because given enough time and opportunity, bacteria will develop one way or another to overcome your antibiotic pressure. But I think in this case, we appear to have given the glycopeptide a secondary mode of action. The first mode of action of all glycopeptides is, of course, binding to the cell wall precursor, lipid 2. And I didn't mention that. I won't go into the details there because that's pretty in-depth stuff. But suffice to say, we think we've added a second mechanism to these compounds, and that can also presumably make it more difficult for bacteria to adapt. They'd have to, in principle, overcome maybe 2 different mechanisms to do so.
Yeah, very interesting. Thank you for that explanation. So we have a question from Julia, and the question is, how do you think that a positive charge affects the performance and pharmacokinetics of the antibiotic? What was the rationale for introducing the positive charge? So it's linked to what we just discussed.
Yeah, I think we've kind of touched on that already in the answer I just gave. As I say, I think that the strong positive charge likely has a benefit in terms of driving or stabilizing the interaction with the negatively charged bacterial membrane. So that's one side of it. And then on the other side, why do we have better PK than you do with these other ones? Well, I think indeed maybe that strong positive charge also helps you from having these very high protein binding interactions that lead to these excessively long clearance times. So it's almost a 2 birds with 1 stone type of thing, as we say in English.
Yeah. Poor birds. Final question from Rob Williams is, what is the new antibiotic target? Is the DALA dipeptide, or is the lipid 2 like a vanco? Or diala, sorry, I meant to translate.
Yeah, no, well, hi Rob, thanks for the question. I think it's I'm trying to think what mechanistic studies we've done. We know, for example, that lipid 2 will antagonize the activity of these compounds. But of course, this is working with wild type lipid 2, which also terminates in the dealanine motif. So I hesitate to say it's not the diala motif. It's certainly lipid 2. I can say that. And I think now that I remember it, Emma would be able to confirm it if she was here, but we have, I believe, tried to run the antagonization experiment with the mutant dealanine DLAC terminated lipid 2, and that does not antagonize the activity of our compound. So that's pretty strong evidence that it's binding to the diala form of lipid 2 and not the diala DLAC form. And then you say, well, then how does it kill the vancomycin resistant strains? Well, there's some discussion there, but in the case of the Van B type organisms, probably vancomycin resistance is simply not turned on by our compounds the same way it is by vancomycin itself. And if you look at our data for the Van A type, we do acknowledge that our MICs are slightly higher, and that's probably a reflection of the reduced binding to the diala-DLAC form, because in Van A, the diala-DLAC form is more constitutively activated. So I think that answers Rob's question.
Very well. There's time for more questions after the final speaker, but for now, Professor Martin, I thank you very much for your contribution. We will now move on to the second speaker of today. This is Professor Jan-Willem Veening, who is a professor at the University of Lausanne. He obtained his PhD from the University of Groningen in the Netherlands and his postdoctoral training at the Center for Bacterial Cell Biology in Newcastle, in the UK that is. In 2009, he established his own research and teaching group in Groningen Biomolecular Sciences and Biotechnology Institute at the University of Groningen. And in 2016, he moved to the University of Lausanne as a full professor. The Veening Lab uses systems and synthetic biology approach to study chromosome segregation, mechanisms of antibiotic tolerance, and noise in gene expression in the important opportunistic human pathogen Streptococcus pneumoniae. And today, Professor Veening will talk to us about fighting the spread of antibiotic resistance with bacterial competence inhibitors. Professor Veening, welcome, and the floor is all yours.
Yes. Okay. I hope you can hear me.
Yes.
Yeah, great. Yeah, so thanks for the nice introduction and the invitation to speak at this webinar. So today I will talk about pneumococcus, another gram-positive organism, very interesting organism. It sits in your nose where it's sort of harmless, where it eats off the mucus produced by your epithelial cells, and its natural life cycle is basically spreading from one nose to the other. And this is sort of a very harmless life cycle, but it can also enter otherwise sort of sterile niches like the lung, or let me see if I can get this to work out. Like your lung or your blood or your brain or your inner ear. And there it can cause really serious diseases. And every year more than 1 million people die because of pneumococcal infection. So pretty interesting organism. On top of that, we also see, of course, an increase in antibiotic resistance in this organism. And quite importantly, also displacement of vaccine serotypes. So in the developed world, we have access to pneumococcal vaccines. They target the capsule, the sort of sugar layer around the pneumococci. But we see new strains taking over the niche of strains that are covered by the vaccines. And here are just some numbers for drug-resistant pneumococci and the costs and death they invoke in the United States. And what is quite peculiar about pneumococcus is that the serotype switching and antibiotic resistance is mainly driven by competence development. So here's just some numbers from a recent study where they sequenced over 20,000 pneumococcal isolates and where they find a very strong association between increased recombination and antibiotic resistance. So what is this competence development? Well, many human pathogens can become competent for DNA uptake. This is a developmental program that can be stimulated. In the case of pneumococcus, for instance, antibiotic therapy can activate this developmental program. And once a bacterium is competent, it's able to take up free DNA from the environment And then, for instance, imagine if this is a coding for an antibiotic resistance marker, you end up with an antibiotic resistant bacteria. Or this could be, for instance, a new capsule locus, and the bacteria would produce a different capsule not included in the vaccine. So this basically, we thought this is very interesting. We've just heard from Nathaniel that it's pretty hard actually to come up with new antibiotics. You have to do very advanced chemistry, but just screening new compounds would, well, people are still doing that, but it's very hard to come up with completely new different antibiotics with different mode of actions. So one idea that we and others are exploring is, can you not look for compounds that do not kill the bacteria, but perhaps prevent the spread of resistance or make them less virulent? So, and the advantage of such, for instance, anti-evolution drugs could be that you do not have this selective pressure, therefore no resistance development. So there's a couple of antivirulence drugs on the market for a couple of bacteria. And I think this is also an interesting avenue to explore. So we were wondering, can we find such drugs for pneumococci, specifically for competence development? So just to give you a quick, molecular biology overview of how competence development in pneumococcus is regulated. At the heart of the pathway, you have this sort of classical two-component regulatory system where you have a small peptide that is exported by COM-AB. Once it's outside the cell, we call it competence stimulating peptide, or CSP. Once there's a lot of this peptide around, when there's enough quorum, so this is a quorum sensing system, this peptide can be recognized by the histidine kinase COM-D, which then autophosphorylates, donates its phosphate to COM-E, a response regulator that then dimerizes, and it can activate a lot of genes, including its own operon. So this creates a positive feedback by which all the cells in the population will become competent. It also activates a gene called COM-X, which encodes for an alternative sigma factor, which then switches on the genes required for DNA uptake. And then this can lead finally to, when there's DNA available, to Rec-A-dependent homologous recombination. So just to give you an idea of how efficient this process really is in pneumococcus, this is work done, some single cell analysis on this process by a former postdoc in the lab, June. He made two strains, one where we have a fusion with a red fluorescent protein where there's a stop codon, and another one which is identical, but it doesn't have this stop codon. And the idea here is that if this strain becomes competent and we give it DNA of this strain, it will start producing red fluorescence, which we can then track under the microscope. And here's just such an experiment where these cells were given a piece of DNA, and then we can follow light, and at one point, indeed, some cells start to express the red fluorescence. And we can quantify this. This is a sort of lineage tree. So here you have the original cell, and you can see half of its lineage took up its DNA and became transformed and now expresses this allele. So this is very efficient. June took it even further, where he gave cells 3 different pieces of DNA, so 3 different colors, and you can see all kinds of variations here. They become blue, yellow, or red, 2 colors, or even 3 colors, just to show that recombination and DNA uptake is very efficient in pneumococcus, and you can imagine that this can be a main driver for antibiotic resistance development. So this slide is sort of 10 years of competence work in my lab, and this shows that certain antibiotic can activate competence. And what we do here is we use a competence-specific promoter that drives firefly luciferase, and then we can, in a microtiter plate, we can examine whether cells become competent. Here you see the growth. This is, for instance, ciprofloxacin at sub-MIC concentrations. When cipro is not present, they do not become competent, but in the presence of this antibiotic, they become competent. And we found basically 3 different mechanisms by which certain antibiotics can induce competence. I don't have the time to go into this, the mechanisms of these drugs, but yeah, we have antibiotics that involve affecting DNA replication, cell wall, or aminoglycosides, so protein synthesis. But this just shows that in the case of pneumococcus, certain antibiotics can induce potentially the spread of resistance. So we thought if you have compounds like these antibiotics that can activate competence, we might also be able to look for and find compounds that can inhibit competence development. So that's what we set out here in a sort of high throughput screen, where we screened a lot of drugs, human drugs, FDA approved drugs, and we were looking for compounds where we would not perturb growth, so we wouldn't have selection for these compounds, but where competence would be well, not activated, so to say. So this is how typically these kind of results look like. So here are some of these potential competence blockers, or COM blockers as we call them. So here on the x-axis you have the growth. So the addition of these COM blockers do not really perturb growth. And here on the y-axis you have luciferase production, so competence development, and they do not produce competence anymore. So we were able to find quite some of these COM blockers. So the next step was, okay, do they actually also prevent then horizontal gene transfer? And to address that question, we made 2 strains. So they are genetically identical, except for a different antibiotic resistant marker at different positions on the chromosome. So this one is tetracycline resistant, and this one is kanamycin resistant. You can grow them together. They will kill each other, so to say, and take up each other's DNA. And you will find, if there's a horizontal gene transfer, bacteria that are both tetracycline and kanamycin resistant. So here's one such in vitro experiment where we grow pneumococci on top of human epithelial cells, so sort of mimicking what's happening in your nose. We inoculate with these 2 different pneumococcal strains, and in the presence, well, in the absence of COM blockers, you have very high horizontal gene transfer efficiency, so 1 in 10,000, so to say. And in the presence of very low concentrations of competence blocker, we absolutely have no horizontal gene transfer anymore. To take this one step further, we wanted to see if this could also work in vivo, so in a mouse model. So here, the team of Birgitta infected mice with these 2 strains, and mice were also injected either with or without or with a COM blocker, in this case proguanil, and then bacteria were collected from the mice and plated on all the combinations of antibiotics to see if there was horizontal gene transfer. And that's what you can see here. If mice were not treated with the COM blocker, you have quite efficient horizontal gene transfer, and every dot is a mouse, I should say here. And when they were also treated with a competence blocker, you have a lot of mice that have no horizontal gene transfer events recorded at all. So to wrap up a bit, I'm not going into too much detail, but what we found in this study published this year is that competence development is very sensitive to the protomotor force. And this is because the export of the CSP, the competence stimulating peptide, is very sensitive to even the mildest perturbation of protomotor force. And what happens if you have a COM blocker that at sub-MIC concentrations already perturbs the protomotor force, you have less production and export of CSP, and thereby this positive feedback loop never kicks in, and thereby sigma X is never produced, and they never make the proteins required for DNA uptake. So that's the story in a nutshell. With that, I'd like to thank my team. I showed the pictures of the people who did the work and the collaborators and the funders, and I'd like to thank you for your attention.
Thank you, Professor Veening, for a very interesting talk. Regarding I mean, it's really interesting, but the question I have, how would you put it to use? Would this something be like you can apply when a patient is already sick, or how do we get rid of the resistance that's already there and circulating in the population? How do I see that?
It's a tricky point, right? Because it wouldn't help if you are already sick with a resistant pneumococcal strain. So you would have to think sort of as the way we think it might be usable. I mean, for us, it's sort of very interesting from a molecular biology point of view. But of course, you could think about would this actually be a feasible approach in this case? But one approach I could think of, imagine people that get very regularly antibiotic therapy, like cystic fibrosis patients. They very often get antibiotic treatment, for instance, to combat the Pseudomonas infections. And what is happening actually, for instance, Astreonam sometimes is prescribed. So this would not at all kill pneumococci, so the pneumococcal microbiota, but it would drive evolution in their pneumococcal microbiota. So you could imagine that in these patients, you might add a competence blocker at the same time, just to prevent resistance development in the future, because very often they might die because of a pneumococcal pneumonia after all, that cannot be treated anymore by an antibiotic resistance. But yeah, it's hard to sell this. I think money-wise or interestingly wise, because it's sort of a vaccine for antibiotic resistance. So yeah, people would not be so interested, I guess, to take that.
I wouldn't say that. I think all approaches feasible should be investigated in this urgent matter. So I think, I mean, it's amazing work. I particularly liked your coloration of the bacteria in 3 colors. I thought that was really cool, actually. And I guess it's also useful for contact tracing when you have a patient with antibiotic resistant bacteria. Because particularly with meningococcal, for example, you do contact tracing. Would there be a possible?
Yeah. Well, I don't really see that point. But you mean if you could, you're still thinking about blocking competence?
Yeah.
Yeah. No. Well, I think I can see a point with for patients that really are heavy antibiotic users, and these compounds that we found are very specific for the 2-component system of pneumococcus. But you could imagine also looking for compounds that would block, for instance, plasmid conjugation. What's a main driver for antibiotic resistance in gram negatives, for instance. So I think the concept of looking for these kind of molecules to reduce the spread of resistance is interesting. Just the implementation is perhaps more difficult. But I think for a subgroup of people that are really, heavy users, where in the end they do get resistant infections. It could be an interesting approach.
Yeah. And then a question from Suzan is, do the competence blockers work similarly on all serotypes?
Yeah, so I think that's a good question. So we screened a lot of different clinical isolates. So the competence system itself is very conserved. So it's really part of the biology of pneumococci. Pneumococci do not have a SOS response like most other bacteria, but they use the competence system as their SOS response. So all pneumococci that we've tested are sensitive, even some Streptococcus mitis, for instance, which is also commensal in your mouth and nasopharynx. So so they work on all serotypes so far.
Very good.
Yeah.
Okay, that's really interesting. Thank you very much, Professor Veening. I think now we're going to switch to our third and last speaker of today. There will be more opportunity for questions in the discussion afterwards. So please keep submitting your questions via the chat function. So now we're going to switch to Professor Suzan Rooijakkers. She's Professor of Microbiology at the University Medical Center in Utrecht. She obtained there her PhD as well, and she did her postdoc in San Diego. Professor Rooijakkers is interested in understanding the molecular interplay between bacteria and the human immune system. And the focus is to understand the mechanisms of bacterial killing by the human complement system and develop immune therapies to tackle infections with antibiotic resistance bacteria. Professor Rooijakkers, welcome. Thank you. And the floor is all yours.
Okay, thank you. Can you see my screen?
Yes, we can.
Okay, great.
No, we cannot. Sorry.
Okay.
I see you, but not your screen.
Now?
Yeah, brilliant.
All right, great. Well, thanks very much for the introduction and of course also for inviting me for this webinar today. Yeah, so as was already introduced, my lab is mainly interested in understanding the interplay between bacterial pathogens and the human host. And for many years, we have actually mainly worked on understanding the basic mechanisms of these interactions. But in the past 5 years or so, we have also looked more into how we can use this information to develop therapies. And then we're mainly looking into antibodies as potential alternatives for antibiotics. And basically this work was mainly started because pharmaceutical companies or antibody companies expressed interest in my work. And they said, well, maybe we can work together and see whether also antibodies could be used as therapies in bacterial infections. And I think the main reason why pharmaceutical companies are now again interested in treating infections, there are basically two reasons. Of course, the first reason is that there is an enormous problem with drug-resistant bacteria. And the fact that we're running out of antibiotics really asks for different approaches, and antibodies could be one. Another important reason to look into antibodies is that they have become very successful in the treatment of cancer and also autoimmune diseases. So that's another reason why it's interesting to look at this. So when I think about antibody therapies against bacteria, I think it's important to realize that antibodies can have multiple effects. So in principle, so in virology, antibodies are used to block the invasion of viruses into host cells. For most bacteria, that of course doesn't work because a lot of bacteria have an extracellular lifestyle. So we're mainly looking into these three mechanisms. So antibodies can be used to block adhesion of bacteria to host cells. And a lot of antibodies that have been developed in the past actually neutralize bacterial virulence factors like toxins. Then another important strategy is to develop antibodies that can actually activate your own immune system. And we mainly look into this last part because I think that this is actually the only way by which an antibody can kill a bacterium. So if you look at these two strategies, these are mainly virulence reducing antibody strategies. But if you have an antibody that can activate the immune system, then the antibody also has the potential to kill bacteria. So what is an important requirement here is that the antibody binds to the bacterial surface, as is indicated in this nice image. And so the binding occurs via the variable domain of the antibody molecule, which is indicated in here. And then the constant part of the antibody needs to interact with other parts of the immune system. And for the defense against bacterial infections, most importantly here is to recruit neutrophils, phagocytes, to the site of infection and induce the phagocytosis of the bacteria by immune cells, because immune cells have the capacity to kill the bacterium. Then another thing which we think is really important is to activate the complement cascade, which will also help the immune system to kill the bacteria. So a little bit more about complement. this is an important part of our innate immune system. Actually, it's a large family of proteins in our blood that can be activated in response to a bacterial infection. And I'm not going to go through all the details of this complicated system today, but it's most important to recognize that in order to activate the complement cascade on a bacterial surface, an antibody needs to bind to the bacterial surface, and then a whole cascade of events will start. And that cascade then leads to the labeling of the bacterial surface with complement proteins. So it's basically the first complement protein, which is called C1, that binds to the antibodies. Then you have this complement reaction, which labels the bacterial surface with molecules which are called C3b. So it's sort of like a protein coat around your bacterium, which is really important for phagocytes to be able to recognize the bacterium well and also eat the bacteria and kill it intracellularly. So we can measure this nicely in our lab. And this is actually a movie that was generated by Dani Heesterbeek, if I can start it. No, it doesn't work. Maybe like this, yeah, okay. So this is a glass slide that's coated with complement oxalized bacteria, and you see that these neutrophils start eating these bacteria. So here you see it again. So, and then you mainly think, well, people who had some immunology background, they think, well, neutrophils can engulf bacteria by themselves, or maybe the antibodies can do that. But from our experience, we actually know that the labeling of the bacteria with complement is really important. So here we quantify phagocytosis in flow cytometry. And basically you see if you just have the neutrophil and the bacteria, then the uptake is really not efficient. Then if you only add antibodies, you trigger Fc gamma receptor mediated uptake, but still not a lot of bacteria go in. Only when these antibodies then activate complement, then you see that this largely enhances the uptake of the bacteria by the neutrophil. So I hope you're now convinced that, that it is important for antibodies to activate the complement system. But the thing that we really don't understand yet is what are now potent antibodies, because we have, you can have antibodies against different parts of the bacterial cell. But the key question really is what is now an antibody that can potently drive this complement cascade and the killing of bacteria. So an important piece of information actually came from work that was done at Uithof. And it's basically collaboration between Genmab, an antibody company, and a group of Piet Gros. And they basically looked into how antibodies can activate this complement cascade. So here on the left, you basically see the old textbook view. So basically we thought, well, there are 2 antibodies are enough to recruit this C1q molecule to the surface. And these antibodies don't need to have a certain organization. However, in this hallmark study, they showed that actually antibodies need to come together on the surface in order to form the ideal docking platform for this C1 molecule. So basically, thanks to the COVID crisis, we can now say that we know that antibodies don't like social distancing. They actually want to come together on the surface of the cell, because that's the ideal situation to activate complement. And a couple of PhD students in our lab made a nice movie to illustrate this. So every student represents an antibody, and then here they show how they need to come together. So the Fc tails of the antibodies need to come together. And now this is the perfect platform for the C1q molecule to dock onto. All right, so we have the structural information about how these antibody clusters should look like. So this is the structure of these IgG hexamers, as we call them. So basically, this is one antibody, and the antibodies come together via FC-FC contacts. And this is important for the clustering on the surface. Now, if you look more closer into the amino acid sequences that mediate this FC-FC contacts, you can also start designing antibodies that are better in making these hexameric structures. And this is something that Genmab has done in the past for antibodies against tumor cells. So basically what they did is they made a bunch of different mutations here in the FC-FC contact region, and they found that these two mutations in the FC tail of an antibody can enhance complement-dependent killing of tumor cells. So together, we started looking into whether this could also work for the development of better antibodies against bacteria. And then today I'm going to show you an example of work that we did on Streptococcus pneumoniae. And I was confident that Jan-Willem would very nicely introduce this bacterium, so I don't have an extensive introductory slide. So what we actually did in this study is we looked into whether we could improve antibodies against Streptococcus pneumoniae capsule. And actually, how do we do this? Well, basically, we just went into the databases and looked whether we could find the variable sequences of antibodies against Streptococcus pneumoniae. And basically, we found one antibody that's directed against capsule serotype 6, and it's an antibody that's specific against the capsule 6. And we cloned this antibody in expression vectors. So this is something that we do now on a regular basis in our lab. So basically what you do here is you take the variable sequence of the heavy and the light chain, and then you can put that onto any constant region of an antibody that you like. So in this case, we have the variable domains recognizing the capsule serotype 6, and then we have the constant domain of a human IgG1. And then we have 3 variants in the FC tail. So we have a wild type antibody, but then also antibodies that have this single amino acid mutation that was reported to enhance the clustering. So this work was mainly done by Lera Aguinagalde, who was a postdoc in my lab. So what we did is, of course, first we confirmed that these different antibodies could bind to the surface of encapsulated Streptococcus pneumoniae. And you see here that basically, if you modify the FC tail of the antibody, then the binding is still the same. So that doesn't affect the binding. So now the question, of course, was how do these antibodies now activate this complement system? So then we add complement into the equation, and then this cascade will start. And now we're going to look into how many C3b molecules are deposited on the surface. And actually the effect was very dramatic. So what we saw is that the wild type anti-capsule antibodies could hardly induce complement activation on the surface of these bacteria, while these hexabody variants showed a very potent C3b deposition. These are the results for serotype 6b, but we have similar data with this same antibody on 19A strains. Okay, then of course, important question was whether this enhanced C3b deposition also leads to enhanced phagocytosis and killing, and it did. So this is an experiment where we measure phagocytosis of Streptococcus pneumoniae by human neutrophils. And now again, we see that the wild type antibody has a very poor capacity to induce phagocytosis, while these 2 hexabody variants are very effective in doing so. And then when you look into the killing of the bacteria, then we also see that wild type antibodies have a poor killing capacity, and at 1 microgram per ml, we see especially that this hexabody variant is very potent in killing. So we couldn't resolve any colony on our plate, but also this variant is better. So we conclude that the enhancement of FC-FC interactions by modifying monoclonal antibodies can improve the phagocytosis and killing of Streptococcus pneumoniae. So now this is a nice proof of concept on Streptococcus pneumoniae, but people who know a little bit about my work also know that we have a lot of experience with Staphylococcus aureus, which has been introduced by Nathaniel already. And we also wanted to know how antibody activation on staph can be turned in efficiently. So I think needless to say is that antibody therapy for Staph aureus is really needed, because this is the leading cause of hospital-acquired infection, and also there is a lot of antibiotic resistance associated with it. Also for staph, we don't have effective vaccines, but we also know that staph produces a lot of virulence factors that can actually block the immune system. So we weren't so sure whether antibody therapies can actually work on Staph aureus. And of our particular interest was actually the main virulence factor of Staphylococcus aureus, which is staphylococcal protein A, which is of course a famous IgG binding molecule that is well known for its capacity to bind to the FC region of IgGs. And staphylococcal protein A is normally attached to the cell surface of Staph aureus, but it's also secreted into the extracellular milieu. So the protein has a cell attachment region, and then it has 5 very similar domains that can all bind to IgGs. So in the past, it has been suggested that by binding to the FC region of IgGs, that protein A can block phagocytosis, but the real mechanism of this was not really clear. And when we looked into the crystal structure of one of these SPA domains in contact with IgG, we actually noticed that protein A exactly binds at the same spot where antibodies need to come together to form these antibody platforms. So obviously we started wondering whether maybe Staph aureus evolved this protein specifically to block, to prevent this IgG hexamerization process. So we started a collaboration with the group of Albert Heck, also in Utrecht, and we started doing mass spec experiments. So basically these experiments were done by Maurits den Boer, who's a PhD student in the group of Albert. And what Maurits did, actually, he started looking into hexamer formation in a kind of an artificial system. So we here have a mutant of antibodies that can form hexamers in solution, which normally doesn't happen because this is really a cell surface thing. But with this mutant, you can form hexamers in solution, and these hexamers you can measure in native mass spec. So here we see that with this mutant, you have hexamers, but also monomers. Now, when we add soluble protein A or just a single domain of protein A to these artificial hexamers, then we see that we can only resolve the monomer, and the hexamer is no longer there. And this happens both with a single domain and also with the full domain. And then as a consequence of this, we can also see that C1q can no longer form a complex with these IgG hexamers. So protein A blocks the formation of IgG C1q complexes on the surface.
Professor Rooijakkers,
yeah?
Sorry to interrupt you, but we are a bit tight of time. Could you wrap up slowly?
Yes. So basically we showed that on the surface of Staph aureus, so if you have monoclonal antibodies that recognize the surface of staph, in this case it's an antibody recognizing maltodextric acid, then we see that protein A blocks complement activation by these antibodies. And what is really interesting here is that we know that protein A does not bind to IgG3 because IgG3 has a single amino acid mutation that basically prevents the binding to protein A. So the simple question here was, if we now modify an IgG1 antibody against Staph into an IgG3, can we then activate complement in the presence of SPA? And that's indeed what we see. So if you modify anti-Staph antibodies from an IgG1 into an IgG3, then you see that protein A can no longer block complement activation. And more importantly, we also see that if we look into the killing of Staph aureus, that IgG3 molecules really have a superior capacity to induce the killing of Staph by human neutrophils in the presence of protein A, while this is blocked in case of IgG1. Okay, so we conclude that protein A is an immune evasion protein that blocks IgG hexamerization, and that IgG3 antibodies have a superior capacity to induce killing. And these are the people I would like to thank, also our collaborators and our sponsors. Thank you.
Thank you very much, Professor Rooijakkers. That was really interesting, and I can assure you that, as far as I'm aware, very few people are enjoying social distancing as well. I also really like your department. Seems like a very much fun. And I was wondering, because your students were also dressed up, which dress code resembled IgG3 to be the most potent?
Good one. I think these were all IgG1s.
Considering the time, there's a few questions from your colleagues.
Yep.
I would like to start with a question by Professor Martin. What do the antibodies recognize and bind to on the E. coli cell surface? Is it LPS? And can this interaction also be enhanced?
Yeah, so my talk was about Staph aureus and Streptococcus pneumoniae.
The video was E. coli, wasn't it, or not?
Which one, with the neutrophils?
I thought so, but maybe I'm wrong. Yeah.
Oh, yeah, that's true. So basically that is a pooled serum. So they will recognize surface structures on the surface of E. coli. So these were not monoclonal antibodies, but polyclonal. So anything on the surface that activates complement is recognized.
Do we have the way that you do, though, for the gram-positives, specific antibodies that engage and recognize specific targets on gram-negative cell surfaces?
Very little. So we wanted to repeat this for E. coli, but there's only one antibody known that can bind to LPS. So there, the route is much further. We probably first need to identify them before we can do all these tricks.
Okay. Yeah. Some nice opportunities to collaborate too. We have some chemical tricks for giving things more affinity for gram-negative cell surfaces. So
yeah.
Thanks.
Yeah, but there are actually surprisingly little antibodies sequenced for E. coli. So there are antibodies, but they're not sequenced. Yeah.
Okay, thanks.
Yeah.
I have a question, Suzan, for these hexameric mutations. Does it mean that will it cause a problem when administering, that it might aggregate and not, distribute very well?
Yeah, no, this is, so basically this is designed to only form hexamers on the cell surface. And I think GenMap has done all these safety studies, like if you have very high concentrations of these modified antibodies, then they will still not form hexamers in solution. That's definitely not what you want. Yeah. And
then I understand it correctly that for pneumococci, you would need for every serotype you would need a separate antibody. Yeah. So look for Staph. We can probably target something that's really conserved on all Staphylococci, like waltacoric acid. But for pneumococci, if you really want to target a certain serotype, then it needs to be against that serotype. So that is an issue there. Yeah. Oh, very cool.
So thank you very much for your very interesting presentations. Given the time, we don't have much time for a panel discussion, unfortunately. But to all the listeners in, dialers in, and those who are going to watch this later on YouTube, you can send us still more questions now via the chat function, or later you can email them to us, and we'll present them to the speakers, and then we'll get back to you. That is the idea. Then for December, there will not be a webinar. This is the last one of 2020. And hopefully 2021 will be a better year, and we'll start in January with new series. What we will do in December is we will send you all a questionnaire asking you for feedback, but also for ideas. What would you like to see as the next topic for our NCOH webinar? And you can send all the information also on our website, or you can send an email to info@ncoh.nl. And I think that with that, Professor Martin Veening and Rooijakkers, again, thank you very much for your time, for your very interesting presentations. And we wish you all a very happy festive season with that.
Right.
Thank you very much.
Thank you.
And with this, the webinar is finished. See you in 2021.